Hi everyone! In this video we are going to talk about the metabolism of nitrogen, and to be more specific, about amino acid metabolism, deamination, transamination, excretion, and everything that has to do with nitrogen in our body. If you are interested to know more about this, or you're just going to fail your next partial test, don't worry. Just keep watching and we will explain you in a few seconds. Now, let's get started. Nitrogen, as we all know, is an element found in our body. But did you know how important it is for us to survive? This element is a fundamental component of amino acids, which are the molecular building blocks of proteins. The human body needs nitrogen to make proteins and amino acids. We can find these structures in muscles, skin, blood, hair, nails, and DNA. In this figure, it is shown the general structure of an amino acid, which consists in a carboxyl acid group, an alpha carbon, the R group that varies for every amino acid, and of course the amino group that contains the nitrogen. Although nitrogen is abundant in the environment, humans cannot directly use it from the air or soil, but instead we depend on microbes and green plants to convert it into a form our body can use. We obtain nitrogen from our diet, for example, when we eat fish, red meat, legumes, nuts, egg, and other dairy products, we're absorbing an important amount of nitrogen. Our body is constantly recycling nitrogen from amino acids. This element can also be used to make other types of compounds that are in proteins, such as heme in hemoglobin, which carries oxygen in red blood cells. Did you know that while many other organisms, even simple ones such as Escherichia coli, can make all their amino acids from scratch, we possess synthetic pathways for only 11 out of the 20 standard amino acids? The remaining ones must be obtained from the diet, and they are referred to as the essential amino acids. This brings us to a very important topic, the amino acid metabolism. The catabolism of amino acids is an expensive task for our body to do, talking in terms of energy. This process accounts for 10 to 50% of humans' body energy production. The liver is the major site where nitrogen is metabolized and excreted via transamination and deamination in order to form urea. Because of the amount of nitrogen produced, the metabolism of this element can be potentially toxic for us. There are 4 different amino acids that have an important role in nitrogen metabolism: glutamate, glutamine, alanine, and aspartate. Glutamate and glutamine are essential because they act like a collection point of amino groups. In the cytosol of the hepatocytes, the amino groups of the majority of amino acids are transferred to the alpha-ketoglutarate and form glutamate. Then the glutamate is transported into the mitochondria where the amino groups yields to form ammonia. The excess of ammonia generated by most of the other tissues become the aminic nitrogen of glutamine, which passes to the liver and then to the mitochondria of the same. The first step in the catabolism of most amino acids, once they have reached the liver, is the elimination of alpha amino groups, catalyzed by an enzyme called aminotransferases or transaminases. In these transamination reactions, the alpha amino group is transferred to the alpha carbon atom of the alpha ketoglutarate, leaving the alpha keto acid corresponding to the amino acid. In these reactions, there is no direct deamination because the alpha ketoglutarate is aminate while the alpha amino acid is deaminate. Deamination reactions occur in the mitochondria and are catalyzed by an enzyme called L-glutamate dehydrogenase. This enzyme is the only one that uses NAD+ and NADF+ as an acceptor of reduction equivalents. The alpha-ketoglutarate formed from the amination of glutamate can be used in the citric acid cycle and in glucose synthesis. As we mentioned, the liver is responsible for the majority of the metabolism of amino acids, but it is not the only one. There are some other organs and tissues that can also break down amino acids and then use the carbon skeleton byproducts for energy, and one example of such tissue is our muscle. So, if we start undergoing prolonged exercise or fasting, our skeletal muscle tissue can actually begin to break down branched-chain amino acids in order to form carbon skeleton intermediates, and those are used for energy purposes. But of course, every time we metabolize amino acids, we form ammonium, and as this process continually takes place, we build up a high amount of ammonium that is present inside our skeletal muscle cells. Ammonium is toxic, and so what skeletal muscle cells must do is to be able to dispose of that ammonium, but they lack the appropriate enzymes. Therefore, ammonium must be transported to the liver for disposal. Our body has some ways by which it can get rid of this ammonium from our skeletal muscle. One of these pathways is known as the glucose-alanine cycle, and it's very important to know how it works. To transport ammonium, we must have some type of carrier that transports through the blood to the liver. To let this happen, ammonium must be combined with pyruvate. Pyruvate is obtained from the muscle where we have glycogen storage, and from those glycogen storage we break down glucose into pyruvate via glycolysis. So we generate ATP. That ATP can be used by the cell, and the pyruvate can be used to combine it with ammonium to form glutamate. And then glutamate is transformed into alanine, and the alanine is the one that can be transported out of the cell into our bloodstream, and that is absorbed by hepatocyte arrow liver cells. Now, in the liver occurs the reverse reaction that had already happened in our muscle. Alanine is transformed into glutamate, and then glutamate breaks into ammonium and pyruvate. Our liver uses the urea cycle to basically help our body dispose ammonium. We had already formed a pyruvate in the liver too, and it's here where we undergo gluconeogenesis, because this pyruvate forms glucose, and this glucose is transported back into the skeletal muscles via the bloodstream. An important part of all this explanation is the urea cycle. Why? Because this is a process in which the organisms transform ammonia into urea, so that can be excreted out of our body in form of urine. Remember that ammonia is potentially toxic for us, and that's why it has to be eliminated. Can you imagine our life without this cycle? Maybe not, because we will die before we realize that. Part of the urea cycle takes place in the cytoplasm, and the other part of it takes place in the mitochondria. The urea cycle actually begins inside the mitochondria with the combination of CO2 and ammonia, which produces carbamoyl phosphatase. This reaction requires two ATP molecules and the enzyme carbamoyl phosphatase synthetase 1. After the formation of carbamoyl phosphatase, this combines with amino acid ornithine and leads to the formation of citrulline, and this reaction is catalyzed by the enzyme ornithine transcarbamylase. In the next step, citrulline is transported into the cytoplasm. It combines with amino acid aspartate and leads to the formation of arginosuccinate. This reaction is catalyzed by the enzyme arginosuccinate synthetase, and it also requires one ATP molecule. In the next step, the enzyme arginosuccinase acts upon the arginosuccinate and lyses it to produce two molecules, which are fumarate and arginine. In the last step of urea cycle, the enzyme arginase acts on the arginine and lyses it to produce urea and ornithine. Ornithine is utilized back into the urea cycle. Whereas urea is a soluble compound, it dissolves into blood and is transported to the kidneys to be excreted in form of urine. Now, just keep in mind that urea cycle is connected to the citric acid cycle by the aspartate arginosuccinate deviation. And, as an interesting fact, a healthy person excretes around 10 to 20 grams of ammonium per day. A dysfunctional enzyme of this cycle can result in hyperammonemia and related diseases. Most common symptoms of urea cycle disorder are vomiting, coma, and seizures, and in this disease, kids are the most affected. In adults, high levels of ammonium in blood can result in a brain edema, which in simple terms is liquid accumulated in the brain. We have reached the end of the urea cycle, so it is obvious that we have talked about how it is expelled, not only in humans, but also in other species. The way of excretion depends on each species. Ammonia as ammonium ion. The ones that excrete ammonium through this one are called ammoniotelic animals, which are in their majority aquatic vertebrate, fishes, and amphibian larvae. Urea. The ones that excrete ammonia through this one are called urotelic animals, which are in their majority terrestrial vertebrates and sharks. And the last one is uric acid. The ones that excrete ammonia through this one are called urotelic animals, which are in their majority birds and reptiles. As mentioned before, our body have two types of amino acids, non-essential amino acids that can be synthesized by the body, and essential amino acids which cannot, and have to be provided from the diet. The non-essential amino acids are glycine, alanine, serine, asparagine, aspartic acid, glutamine, glutamic acid, proline, cysteine, tyrosine, and arginine. The essential amino acids include valine, leucine, isoleucine, phenylalanine, tryptophan, methionine, threonine, lysine, and histidine. Now, if we count the amino acids, we can see that in total there are 20. These are the ones that our body can process to make proteins. Each of them have a catabolic pathway that at the end converts to form just 6 principal products that enter to the Krebs cycle. Depending on their metabolic phase, amino acids are referred to as glucogenic and ketogenic. Glucogenic amino acids are broken down into one of the following metabolites: pyruvate, alpha-ketoglutarate, succinyl-CoA, fumarate, or oxalacetate, which can be oxidized into CO2 and water to generate ATP via the tricarboxylic acid cycle, best known as Krebs cycle. Alternatively, the carbon skeletons from these metabolites can be used to synthesize glucose by the process of gluconeogenesis, hence the term gluconeogenic amino acid. Easy, right? In contrast, ketogenic amino acids are broken down into acetyl-CoA and acetoacetyl-CoA, and they can give fatty acids and ketone bodies in the liver, where acetoacetyl-CoA is converted into acetoacetate, and subsequently in acetone and beta-hydroxybutyrate. This type of amino acids cannot produce glucose molecules in humans. A third group of amino acids are catabolized to both glucogenic and ketogenic intermediates. We already mentioned that amino acids are grouped together according to their final degradation product. Some amino acids can be glucogenic and ketogenic because they are degraded to different final products. To help you understand what has just been mentioned, we are going to focus on the citric acid cycle, especially in the products. Leucine and lysine are exclusively ketogenic because, as we can see, they can only be degraded into acetyl-CoA and acetoacetyl-CoA. Then we have 5 amino acids that can belong to both groups. These are tryptophan, isoleucine, phenylalanine, tyrosine, and threonine. Note that phenylalanine can be degraded to acetoacetyl-CoA and fumarate. The remaining 13 are gluconogenic only. The carbonated skeletons of 6 amino acids become totally or partially into pyruvate. These amino acids are alanine, cysteine, glycine, serine, threonine, and tryptophan. Pyruvate is very important because, as we already know, it helps us to generate glucose, and also it can be transferred into acetyl-CoA.
So this is all for the video today. I hope you guys like it, and thanks for watching. See you next time. Bye bye.